Hi, my name is Li Tai. I am a scientist here at Bina Technologies. I conduct research on cancer analytics. Cancer is a disease of the genome. I'm going to talk about SomaticSeq. It is a machine learning approach that we have developed here at Bina Technologies to detect somatic mutations in cancer. One of the challenges is that tumors are very heterogeneous. There are many different subclones in a cancer. Each of them have a different mutation profile. And some of the mutations have very low frequencies, and that's the reason it's difficult to accurately detect somatic mutations in cancer. Another difficulty is that somatic mutations are actually quite rare. It occurs about 1 per million base pair. I'll describe the methodology of SomaticSeq, which is a machine learning approach that we have developed to accurately detect somatic mutations. We have incorporated 5 different somatic mutation colors to improve the sensitivity of our workflow. And then we use machine learning to filter out the false positives from those call sets to improve the precision. Here is a schematics of the whole SomaticSeq workflow. Each of the colors has a unique algorithm to detect somatic mutations. Some tools are better at detecting rare mutations. Others are better at detecting mutations that has a changing signal between the contaminated normal and the tumor. We will combine those 5 call sets into an ensemble. And for each of those somatic mutation calls, we generate features based on their strand bias, mapping quality, base quality, etc. We use machine learning algorithms to distinguish true positives from false positives based on those features. Here you can see a Venn diagram of 5 different colors. The 5 colors are Mutec, VarScan, Joint SNV Mix, Somatic Sniper, and Vardict. In the center, you can see about 5,000 calls that are agreed upon by all colors. One can use consensus of the colors as a way to filter out the true positives from false positives, but the color themselves do not always have the same performance based on the complexity of the tumor. The machine learning algorithm uses 70 different features. Here we're showing 4 different features, and you can visualize how they can distinguish the true positives from the false positives. If you look at the mean base quality scores on the upper right, you rarely find any true positives that have a base quality score below 30. And the machine learning algorithm will use up to 70 features like this to figure out what is a true mutation and what is a false positive. Here's an example where we use Dream Challenge as a benchmark for our machine learning algorithm. We have used stage 2 of Dream Challenge as our training data. We have built a classifier based on stage 2 and used the results to predict somatic mutations in stage 3. On the left is a histogram of the probability values assigned to each mutation calls. You can see 2 very clear clusters, one around 0 and the other ones around 1. If you zoom into the cluster around 1, you can see that there are calls that's between 0 and 1. But by far and large, the true mutations and the false positives are quite clear. If you look to the right, you can see 3 lines. The green line is the precision, the blue line is the recall, and the red line is the F1 score defined on the graph. So for any probability value you pick, the accuracy, defined as F1 score here, is almost always around 0.95. It's a very accurate approach. Here we're showing a result for Dream Challenge as we have described before. We have generated 4 different settings from the Dream Challenge data. Setting A is straight from the challenge. Setting B is where we artificially spiked in some contamination into the normal to make it a more challenging normal set. Setting C is where we spiked in more normal into the tumor to make the tumor more contaminated. Setting D is where we cross-contaminate. So from setting A to D, the data gets more challenging. SomaticSeq has outperformed each of the individual tools that it has incorporated for both SNV and indels. We have also validated our SomaticSeq using in silico titration. Basically, we mixed two different human genomes and pretend one of them to be normal and one of them to be tumor. We can also mix them at different proportions to simulate tumor or normal of different contamination profiles. We have 6 different settings for in silico titration data. The 6 settings are written on the x-axis of this figure. And 0T50, for instance, means that the normal has 0 variant allele frequency, whereas the tumor, T, has 50% variant allele frequency. It is pure, and the normal is pure. It is the easiest setting that we can create. On the last N2.5, T15, it means that the normal has a variant allele frequency about 2.5% coming from the contamination of the tumor. And tumors only have variant allele frequency of 15% due to contamination of the normal. It is the most challenging set we have created here. And for the 6 settings, the data goes progressively more challenging when you go from left to right. But for each of the settings, you can see that even though SomaticSeq's performance decreases, it's still much better than each of the individual tools that it has incorporated for both SNV and indel. And finally, we have demonstrated SomaticSeq on real data. To predict somatic mutations in real data, the best method is to randomly select a number of mutation calls in your data, validate them for both true positive and true negative, and build yourself a gold set where you can use machine learning to train. Absent of that, we have used Dream Challenge simulated data as a training set and applied that classifier to these whole genome sequencing data. The 2 data sets we have tested here are Colo A29, that is a melanoma cell line, and CLL1, that is a leukemia cell line. We do not know what are the true negatives of those real data. We can only calculate the sensitivity. So in those 2 data sets, you can see that SomaticSeq has predicted a sensitivity that is almost as high as any individual caller that has incorporated, but with a call set that's much smaller than any of those tools, implied that it has a precision that is higher than any of the individual call sets with minimal sacrifice in sensitivity. I have described the SomaticSeq workflow with 5 callers, but it is a very flexible workflow that can incorporate any combination of somatic mutation callers that you want to run.